I'm going to start by telling you a little bit about why we need rapid sequencing for acutely unwell children in diagnostic practice. So 1 in 17 of us will develop a rare disease at some point in our lifetime. 80% of those are genetic, 75% affect children, and unfortunately they're responsible for about 28% of neonatal intensive care deaths pertinent to this talk. So, the rapid whole genome sequencing service for acutely unwell children with a likely monogenic disorder is called the R14 service in England, and it was the first commissioned test as part of the NHS Genomic Medicine service. It started off as an exome sequencing service in October 2019, and more than 2,000 families were sequenced as part of that. In October 2022, it moved to rapid genome sequencing, and since that time, more than 600 children have been tested through that service using short-read sequencing. The approach that's used is called gene-agnostic, hypothesis-free trio testing, if both parents are available. If one or neither parent is available, then we use a virtual panel test. Our current diagnostic yield is 41%, and that's 4% higher than when we were using exome sequencing. So this is a little girl who was diagnosed through the exome sequencing service, Ellie Rose. When Ellie Rose was 6 months old, she unfortunately stopped being able to breathe properly. She lost the ability to walk and talk and spent an extensive time on intensive care. Doctors didn't know what was wrong with her. She had a number of tests, and then she had rapid exome sequencing trio testing with both of her parents, and was diagnosed with a very rare metabolic disorder called methionine synthase deficiency. She was started on treatment. It's a treatable disorder, and immediately started to make progress. I'll tell you more about Ellie Rose in a moment. This is Ruben, who was 7 months in this photograph, but is now 14 months old, from Cheltenham. He was tested through a pilot of our short-read rapid whole genome sequencing before we started in October 2022. He was born well, but at 5 days old, became incredibly unwell, and was readmitted to intensive care at Bristol Royal Hospital for Children, where he had incredibly high levels of ammonia in his blood, potentially lethal, which needed to be filtered. He had metabolic tests which suggested a urea cycle defect, implicating one of 3 genes, including a gene called CPS1, but initial genetic testing and other tests were inconclusive, and in order to confirm his diagnosis, a liver biopsy would have been required, which in an unstable child like that could be potentially fatal. So when we moved from exome sequencing to genome sequencing, we were able to improve the copy number variant calling. We were able to look at previously reported pathogenic variants, regardless of where they are in the genome, so in the intronic and regulatory regions. We were able to look at non-coding genes, and where the clinician is sure about the particular condition a child might have, like there's one gene or a small number of genes, like in Ruben's case, then we can look more extensively across the non-coding genome. But we still can't do everything. So what do we need to do next? I wanted to show you this video because this is Ellie Rose after she had had her treatment. And I think if you were ever doubting why this sort of testing is important, it really does show you why.
Hello there. I just wanted to say a few words about my daughter. When she was 9 months old, she was really poorly and had a chronic breathing condition. She ended up in intensive care for 6 months. But during that time, she had some quite pioneering genetic testing done, and we got a diagnosis. As soon as we got that diagnosis, she was medicated and has made incredible progress. She initially had a tracheostomy, which we battled with for 18 months, but last October the tracheostomy was removed, and now we've got a really happy little girl, and so that diagnosis to us meant everything. And here she is. Say hello, Ellie Rose. Hello.
So shortly before Ellie Rose had that test, her parents were told on Christmas Eve that she had probably not very much longer to live. So if there's anything that motivates us to want to do this testing better and faster and to be able to find more diagnoses, it's stories like that. and then stories like Rubens. So we were able to identify Rubens' second variant. So he had a deep intronic variant that was looked at after manual analysis of the non-coding region of CPS1. That confirmed his diagnosis of carbon phosphate synthetase 1 deficiency. He did not have to have a liver biopsy. He was put on the transplant list. He's now had his transplant, and his parents tell me that he's doing really well and now enjoying eating, which he wasn't doing before. Developmentally, he is entirely normal. So again, a real success story for this type of approach. And I'll just give you a second to read what Rubens' parents said about the experience that they had. So, as I said, stories like Rubens and stories like Ellie Rose make us want to do this better, make us want to find more diagnoses. So what are we missing? We're missing novel non-coding variants outside our current region of interest, so we can't look at all of the non-coding regions yet and prioritize those variants. We wouldn't be able to do that rapidly. We only know what about 7,000 genes in the genome are associated with in terms of human disease. We're not able to look at structural variants. We can't look at methylation abnormalities. Trinucleotide expansions are possible to look at, but we have not yet incorporated that in our pipeline. And for singletons and duos, so when one or neither parent's available, we're limited to a virtual gene panel. We can't often phase variants unless the variants are closer together, and we can't determine whether a variant's de novo or not. And 1 in 10 of the patients that come through the service hasn't got both parent samples available. So what we're hoping is that by using Oxford Nanopore approach, we would be able to tackle these remaining challenges. So if we are able to sequence using long-read sequencing the bits of the genome that are currently intractable, we will discover new genetic causes of disease. If our scientists identify a gene that is a strong biological candidate, or the variant is a particularly pathogenic looking variant, like a loss of function variant in a gene that isn't yet associated with disease, they will put those findings on Gene Matcher. Obviously, if we can find more of those novel associations, then we will discover more new disease genes. We will be able to better characterize structural variants and detect them. We want to be able to reliably pick up SMA. We want to be able to pick up methylation abnormalities, these trinucleotide repeats and phase variants. Long-read sequencing gives us the potential to do almost all of those things. So that, for us, is the next step. I want to give you an example of how long-read sequencing has been used in the development of this approach going forward. So this is an example where we only had the affected child and one affected parent, and 2 variants in the SIL1 gene were identified. Biallelic autosomal recessive SIL1 variants associated with a condition called Marinesco-Sjögren syndrome. That's a complex condition where you have cataracts and neurological impairment, and the child's phenotype was compatible with that. A maternally inherited, clearly pathogenic splice site variant was detected, but there was a second variant detected in the child not present in the mother, a 74 kb duplication on 5q31.2. That variant spanned exons 3 and 4 of that gene. Now, it's very difficult to assign pathogenicity to duplications, as we don't know where that duplicated material is, but long-read sequencing gives us that opportunity. So there were 3 possible options, 2 of which would involve an inverted duplication, and the last of which a tandem duplication. So short-read and long-read sequencing were used together to refine that structural variant, and we were able to see that, in fact, it was a tandem duplication. There was a small inverted section of that duplication, but it didn't involve those 2 exons. So we were also able to confirm that that variant was the paternally inherited variant. So what are the challenges that remain for rapid genomics for critically ill children? We want to make more diagnoses, we want to do them faster, and we want to do it better. And working with Oxford Nanopore and doing long-read sequencing, we hope that we will be able to achieve some of those goals. So that is the PromethION 24 in the NHS Laboratory in Exeter. All of the research and development for long-read sequencing will be undertaken in the NHS Laboratory, so we don't then need to transfer that across to diagnostic service subsequently. I talked a lot today about what's been done. This is actually the team that do all the work. So Andy Parish next to me is the head of the laboratory and bioinformatics and informatics lead for the Southwest NHS Genomics Laboratory Hub and head of lab in Exeter. And Karen Stalls on the other side of me is the head clinical scientist lead of the service. This is the fantastic clinical scientist team that spend many hours of the day and night getting these diagnoses for children. So it's their work really, not mine. Thank you for listening.